Hello everyone. In previous lecture series, we have discussed about the initiation, origin, unwinding, and different factors involved in the prokaryotic DNA replication process. So now we will discuss about the replisome assembly and the elongation process. Then we will discuss the termination process of prokaryotic DNA replication. So in initiation replisome assembly, usually it depends on, in the previous class, previous lecture series, we have discussed about the uh, attachment of DNA A protein, then DNA B, DNA C, and DNA G, which is a primase attached to the origin site of the prokaryotic DNA, and how it acts on it, and how the DNA B unwinds the DNA, how the primase is working on DNA for making RNA, short strand of RNA primer. We already have discussed. So now we will discuss the assembly of this RNA replisome. That means uh, how the helicase enzyme initiated for making the replication fork, and within that fork, what are the recruitments of other enzymes are there, we will discuss in this video lectures. So usually in the uh, prokaryotic DNA process, usually what happens, we already seen that our DNA B and DNA G complex assemble, as in the first picture you can see the complex assemble at the DUV region of the origin of replication, and the DNA G, which is a primase, make an RNA primer. Since we have two strands DNA, two single strand of DNA, and there is also a complementary complex loaded on the DNA strand. So we have one complex overlapping the L repeat of the DUV and the other at the R repeat, which also overlaps with the triose region. And this state is what we call the activated helicase. And the complex of DNA G and DNA B and loaded on the origin is collectively called as primosome. The complex of DNA G and DNA B is called the primose. So primosome, sorry. So following this, the origin is ready for the recruitment of DNA polymerase 3. So we will focus on the DNA polymerase 3 recruitment, which is referred to as the holoenzyme, because the polymerase requires help for the loading, because the polymerase, it needs the loading of its to the strand. The loading needs to occur at the RNA, at these RNA primers, and that are generated by the primase. And specifically what we need is to position the DNA polymerase near the 3' end of the RNA primer, because that is where the DNA synthesis is begin, and then the polymerase will move on the template as the new DNA strand is synthesized. But as we know, polymerase requires help in the loading. Specifically, there are two helper complexes in this process. The direct help in the loading comes from the beta-2 clamp. The direct help in loading of DNA polymerase on the DNA is due to the beta clamp, 2 beta clamp, and this complex directly recruits the DNA polymerase 3, 4 complex at the RNA primer. DNA polymerase 3 is a multi-protein complex. As we know, it has 10 subunits, but the three important ones to keep in mind are alpha, epsilon, and theta subunits. The second helper complex is the beta-2 clamp loader complex, which loads the beta-2 clamp onto the RNA primer. This means that the beta-2 clamp loaders start the process of polymerase loading. So let's start by discussing the clamp loader and how it works and loads the beta-2 clamp. Clamp loader is sometimes also called as tau and gamma complex. Tau and gamma complex, and it has 5 major proteins, and they comes together to form a spiral structure. As we will see that 3 of these 5 proteins have triple A plus domain, which means that they have ATPase domain. These tau and gamma are actually pretty important nomenclature. They refer to two types of they refer to two steady types of form. So we will keep things simple and say that the clamp loader can come in two very fundamental conformation. The basis of the two states come from the difference in the 5 protein. So let's discuss this protein and the structure of the clamp loader. The other 3 proteins are either tau. So the two constant protein in the clamp loader are the delta prime and the delta protein. Here you can see the structure. The other 3 proteins are either tau and gamma proteins. These are the proteins with a ponytail, we can say. So when talking about the 5 proteins, we have 3 tau proteins and 1 delta prime and 1 delta protein. These tau protein have the ATPase domain in them, and for that reason, we call this bottom half of the structure as a ATPase module. Even and for that reason, even though the delta protein don't have the ATPase domain, only the this globular portion, you can see the globular portion in the picture, the globular portion which is a part of this entire top structure, but mainly the globular portion is called the polar module. Okay, so one important thing to keep in mind is that the polar module is sealed and nothing can the polar module is sealed. So things can enter from the bottom, but they can't exit from the module. On other things to notice is there is in this structure is that there is a gap that actually allows, that green color is the gap that actually allows things to enter into the DNA, into the structure, and this is the general structure of the tau complex clamp loader. Sometimes you can also find 2 additional protein on the tau complex. These are the phi and chi subunits. The globular portion, which is the part of this entire top structure, but mainly the globular portion is called the polar module, as we uh, recently discussed. And you can also find the 2 additional protein like your tau complex, protein on the tau complex that are phi and chi subunits. So technically speaking, we could name this entire complex configuration as a tau del 3 delta prime delta chi phi. The entire subunits, entire structure is called tau 3 delta prime chi phi. Since we said that there are two steady states, the other simple state of the clamp loader looks very similar, except that instead of the tau protein, you can find the gamma proteins. The gamma proteins, as we can tell, don't have the ponytail. The remainder of the configuration looks pretty much similar as in the left right hand side. You can see the structure. It does not have any ponytail. So, and this is the gamma complex clamp loader. Once again, we can also write the technical name of the clamp loader. The tau and gamma proteins are product of the same gene called the DNA X. The gamma protein simply lacks this ponytail, and that's because it is a truncated version. The end of the ponytail, which are only present in the tau protein, binds the helicase DNA B. The phi and chi protein helps in the recruitment of clamp loader to the single-stranded DNA because they have affinity for single-strand binding proteins, and the gap, as I said before, helps in the selective entry of the DNA into the clamp loader. The caveat or the caveat remains that the Kolar module is sealed, so nothing can exit from it. The tau contained clamp loader seems to be commonly used clamp loader. The two steady state forms are actually a simplification of something much more complex. In reality, you can have clamp loader where tau and gamma proteins are part of the same. In instance, you can have the clamp loader where you have two tau protein, but instead of third tau, you have a gamma protein and the remainder is a constant. And you can have any variation and combination as long as the tau and gamma proteins add to a total of 3. In any cases, this clamp loader with ATP at the ATPase domain is competent for the beta 2 clamp binding, competent for the beta 2 clamp binding. So the clamp loader is now going to bind the clamp, and it is now only fair that we discuss now. The structure of beta 2 clamp before going into the loading process, it is a homodimer of the beta protein. Since it is a dimer of beta, we call it as beta 2 simply. So simply we call it as a beta 2. So the dimer has a ring-shaped structure, which is, you can see in the picture. The dimer has a ring-shaped structure, which is referred to contain a pseudo 6-fold symmetry. Visually, the beta clamp looks quite boring. You can see it here. And when the ring is open, it is called an open complex. Very creative nomenclature is there. So the other side of the steady state is when the ring is closed, and that is referred to as a closed complex again. And some people think that these two states can coexist in the free form. That may or may not be true. Anyway, the internal edges of the ring have net positive charge. This is just like the helicase, and it helps the ring contact the negatively charged DNA. The predominant view is that the open complex binds the clamp loader. A more interesting way to think about this is that the clamp loader helps maintain the whole structure. So how does this thing happen? A more interesting way, the open state of beta 2 clamp, you can see in the bottom structure, you can see the open state of beta 2 clamp, where the tau complex is our primary clamp loader complex, and now the competency of the clamp loader to bind the beta 2 clamp or to keep it in the open state comes from the fact that the ATP must be bound at the AAA plus domain. Once the ATP is there, the loader can engage with the beta 2 clamp, and the initial docking of the clamp is done by the delta subunits, and then tau subunits come to position that and come to the position of the clamp properly. So now that the beta 2 clamp is bound, notice this gap here, and we discussed the gap a few moments late. We already have discussed it about the gap. See how the clamp itself is also open. As we will see, this gap is perfect for the DNA entry into the AAA plus module. And this AAA plus module, however, in the right hand side, you can see the structure. This AAA plus module, however, has a constraint, and it doesn't like the B form DNA. Okay, but it loves A form of DNA. In case, as we know, B form DNA has its bases oriented at 90 degrees of the helical axis, whereas the A form has its bases at less than 90 degrees. Conveniently, the A form DNA is found in the RNA-DNA duplex, and remember that the primase made a short RNA primer, and this is the perfect setup for the clamp loader and the clamp to bind to the structure. In addition, the single-stranded DNA around this area is loaded with the single-strand binding protein, and so the complex also attract the clamp loader through its phi and chi protein. So how does the loading happens? If we see, then first the clamp loader has to bind the clamp, that means beta 2 clamp, and which keeps the clamp open after binding. Now the RNA DNA duplex is positioned at the open mouth of the clamp loader with the assistance of the phi and chi protein. The entry of the DNA occurs at this gap here, and just for clarity, the single-stranded DNA is pushed to the side, and the single-stranded DNA binding protein interact with the phi and chi protein. At this stage, the duplex, which has the RNA, is just at the mouth of the clamp. The beta 2 clamp now attempts to slide onto the duplex. As a result, the RNA DNA duplex moves into the AAA plus module. Again, for the clarity, when the clamp moves here, what it would looks like. Again, the problem is that there is a duplex inside the AAA plus module, and the 3' end of the RNA primer cannot be pushed to the side, and it can't leave through the polar before it. So we have the single-stranded DNA push out, and the 3' end of the primer stuck inside. This configuration activates the ATPase domain in the AAA plus module. As a result of this activation, the ATP is released. Now this release changes the structural state of the clamp loader and it releases the beta. Since ATP was required for the clamp loader to maintain the open state of the beta-2 clamp, as a result, the beta-2 clamp closes immediately. Okay, as the beta-2 clamp closes, it encircles the DNA-RNA duplex. Since it is a positively charged in the center, its binding is pretty stable. This beta-2 complex in its closed state can initiate the loading of the DNA polymerase, specifically by attracting the alpha core of the enzyme, especially in the grand structure. We can see it here, how all the loader and clamp will be comes together. We can see it here in one coherent. In this picture, we just discussed that the clamp loader loads the beta-2 clamp onto the RNA DNA duplex, and now a polymerase can come and bind into the loaded clamp. Specifically, the alpha core binds associates with the beta-2 clamp. The alpha subunit is the actual polymerase domain for making new DNA. It usually comes with the epsilon and theta subunits. The epsilon is the proofreading subunit which has the exonuclease activity, whereas the theta stabilizes the epsilon onto the alpha domain. We will discuss them in the elongation process, and as the clamp loader after the ATPase activation releases itself from the clamp loader. But we also said that at the beginning, the ponytail of the tau protein binds clamp loader, binds to helicase as well as the DNA polymerase, because as already have discussed that the ponytail of the tau protein binds to the helicase as well as the DNA polymerase alpha core. So the clamp loader also function to position the polymerase at the beta-2 clamp, and it remains intact with the polymerase, and it can also remain in contact with the moving helicase. If the clamp loader has if the clamp loader has 3 tau protein, the tau is usually bound to a spare of DNA, to a spare DNA polymerase of alpha core subunits. This is how we can say that in some cases a new DNA polymerase is needed. The replacement does not take place too long. The clamp loader usually reacquires ATPase at this point, and which means it can also bind a new beta-2 clamp and keep it in the open state. Again, this is a spare clamp which can be used when it requires. The extra polymerase and beta-2 clamps will remain important when we will discuss in the lagging strand synthesis. So technically, in this configuration, we can say that a clamp loader bind to the clamp, the core polymerase and spare clamp. Another strand that goes in the opposite direction. For that, for the sake of completeness, while the beta clamp, you can see it here, the beta clamp and loader with the primosome are present in the L region overlaps on the top complex, whereas R and the triose overlap. The problem now is that the helicase is going to going by the 5' to 3' direction, but the polymerase want to go by 3' direction. Since it makes the new strand in the 5' direction, and as the helicase likewise on the other replisome, the helicase and polymerase are moving away from each other. This is an issue because, as we already have discussed, that the ponytail of the clamp loader binds both of them. So it implies that it must remain stably bound. And therefore, there are two solutions to get around this problem. One is that the helicase moves ahead even before the clamp loading occurs. So the clamp loader cannot bind the opposing helicase at the duplex, which means that while this clamp is being loaded, this helicase has moved to the other strand. The same goes at the other helicase as well. So it also moves ahead before the clamp loading occurs. The second solution relies on the fact that the ponytail are always in the constant exchange state. That means it is not stably bound to the helicase. Therefore, the helicase can go ahead in different direction, and uh, these discussions or this confirmation allows for potential reassociation of the complex to the DNA. Because everything is dynamic and moving, both of these solutions are actually true and give the same final result. So as we already have noticed that helicase which was originally here has moved to the other end. The clamp and the polymerase is still there as expected in the opposite strand. So the the heli now the clamp loader can bind this helicase and reoriented itself, and in this state the helicase is moving in the same direction as the polymerase on the other direction. So by the way, the new strand which is made in the direction of helicase movement is called the leading strand, and we will discuss in this much in details about in the elongation step. The primosome can now make new primers as it moves, and these primers are made. The clamp loader can load the beta 2 clamps on them, and then this polymerase can also be loaded immediately. But the synthesis of DNA will be in the opposite direction to the movement of the strand, and this is the lagging strand. But in general, this is the replisome at the initial step, which is also called the initial replication form, and where you have a bunch of protein, namely the helicase, primase, beta 2 clamp, the polymerase 3, and the clamp loader, which can exist in we can exist till termination process. So let's start discussing. This is all about the replisome assembly process of initiation. So let's start discussing the elongation process. In elongation, as the replisome is moving to 5' to 3' direction, then it will continue in synthesis of DNA to making the newly synthesized strand in leading strand, and in lagging strand there will be a discontinuous replication where primer will be synthesized and then DNA polymerase 3 will add. Then small fragments of DNA is synthesized, then dissociated. This fragment of DNA in prokaryote is called Okazaki fragment, and it is about 1,000 to 2,000 base pair in length. In elongation process, we will discuss the biochemistry of this polymerization. As DNA polymerase 1 recognize the nick in the DNA, it has carboxy terminal domain, and this terminal domain is important for the 5' to 3' polymerase activity, and the clino fragment we can say, and it has also the proofreading activity. So when the Okazaki fragments are there in the lagging strand, so the fragments will be joined, and the RNA primers are removed by flap endonuclease enzymes, and the fragments will be joined by DNA polymerase 1 as it has 5' to 3' polymerization activity. So if we see the structure biochemistry of this elongation process, then we can see the polymerization occur towards the 3' end direction of the growing strand, and here you can see that in the picture that in the intuition of the process by stretching the strand out of a template DNA strand which has a phosphate, and when the RNA primer is laid on the template strand, so here is the RNA primer, you can see it here, just to bound by hydrogen bond, two hydrogen bond between A, hydrogen A and T, and U, and three between G and C. The important things to keep in mind is that the final basis has a free hydroxyl group at the 3' position, so that the now you can see how the substitution will be made, as you can see the DNTPs, uh, and we have 4 of them. DNTPs are there, they all have 3' hydroxyl group and the 5' triose phosphate group. For each reaction or synthesis step, we need one of them. So there is a 25% chances that each one is sampled for given reaction. Given that we have a cytosine in the template and a guanine will be used in the particular synthesis step, because it is the only DNTPase that will fit the polymerase active site and properly base pair with the cytosine on the DNA template strand. In the process of synthesis, the DNA polymerase will try to attach the hydroxyl group of the previous sugar to the phosphate of the incoming DNTPase. The reaction is between the hydroxyl group of the previous sugar 3' end to the phosphate of the incoming DNTPase. As a result, the remaining 2 phosphate are released as a byproducts. This bond that is formed between the hydroxyl and the phosphate is called phosphodiester bond, and this again leaves the hydroxyl available, and the phosphate is available for the next DNTP to be added, just as previous hydroxyl existed. So in the next step, the cytosine will be used because there is a G in the template strand, and in this DNA polymerization will continue till the end. In this simplified view of DNA polymerization, all we want to discuss is that to take away in that form the direction of 5' to 3' comes because the hydroxyl exists at the 3' end, and that is the only place to add a new DNTPase. Now we are ready to now we can discuss the deeper dive into the subject, that we will understand the synthesis by looking at the enzyme which does this function. A specific example will be the C-terminal. By looking C-terminal domain of the DNA polymerase 1, which, as we have already discussed, that clinofragment, it is a clinofragment, and this exodomain, the polymerization function is well conserved into the prokaryote. Therefore, you can also apply the same analogy to the eukaryotic DNA replication process. Here is the structure in the right hand side. You can see the C-terminal domain of DNA polymerase 1, the top left of this structure of the DNA polymerase, and this top right side is called the thumb domain, and the middle portion is called the palm of the DNA polymerase. The bottom side of the structure is the wrist portion. If you can if you may, is the exonuclease domain of the CTD. So this entire thing is like a hand or hand of a baseball globes. We have seen this same structure when we talked about the RNA polymerase. When we discuss transcription, our main focus will be on the top bigger portion or the clino part specifically, and it will not focus on the exonuclease domain. One contrast to note is that the palm domain is largely composed of beta seeds, which help to accommodate uh, the DNA. The important thing is that the palm domain contains the active site for the DNA polymerization activity, and this active site contain two divalent ions, which are usually magnesium or zinc. So when a single strand template comes at the palm domain, it is bent at the palm domain, and the organization is such that the DNA polymerase is recruited at the palm domain, at the 3' and the RNA primer. That means when the DNA polymerase is recruited at the 3' end of the RNA primer, and we have seen in this picture is that this RNA primer as a daughter DNA, because we can think this RNA primer as a daughter DNA because it is a same thing for our understanding that this bending of the DNA is important because it exposes the template for the DNTP addition. So we are now moving to the next picture. In the alpha helix, there are tyrosine, lysine, and arginine amino acid that will become very important for the structure. Another things that this bending of the template, which exposes the template to polymerization, essentially the exposed template is ready to accept the DNTPs. Even more specifically, the bending causes the minor and major groups to become accessible by the active side of the DNA polymerase. And as a result, to as a result, the few to the DNA form B form of DNA in the remainder of the double-stranded part. All this is occurring at the active site present in the palm region. The accessibility is initiated by these three amino acids in the finger helix, where tyrosine plays an important role in contacting the DNA and DNTPs. And lysine and arginine mainly contact the DNTPs. This bending and accessibility makes the polymerase accept a new DNTPs, which can be sampled, and if it is the correct one, the polymerase will use it. So now our focus will shift to the smaller frame where the active site dynamics of DNA polymerase is seen. And this state of DNA polymerase where the DNTP has not yet been confirmed for addition into the daughter strands. That means moving to the palm region, the polymerase will use it. So now our In this state, the polymerase where the DNTP has not yet been confirmed for the addition into the daughter strand is called the open configuration of the DNA polymerase. And when the finger domain becomes accessible, that we mean to say that towards the palm region, and this accessible towards the palm region, and this is about 40 to 46-degree movement. Some articles may say that it moves 30 to 60 degree, but keep in mind that the movement can be different. The point being, the point is there is a movement in the finger domain. It is not important to know how much degree it is bending. The movement occurs when tyrosine respond when it comes in contact with the new DNTPs. And in context of the template DNA that is being bent at the palm region, so let's assume that the next base to be replicated in this tyrosine, this means that the next DNTPs to be added will be the DGTP. So this moves in the finger domain will occur in response to correct DNTPs when it is being positioned against the cytosine on the template sand. So essentially, in the sampling for correct DNTPs of fit in the terms of proper hydrogen bond will cause a sustained move in the finger domain, and now the G and C can properly hydrogen bond. So to emphasize that the point, there is a stacking interaction by the tyrosine on the finger domain, and when pushing the DNTP against the base on the template strand to be copied. And of course, some other weaker interaction like van der Waals forces are also at play here. Let's also complete the picture of the GTP here, since it comes with a sugar and 3-phosphate. And uh, this picture in this slide not complete without the daughter strand, which has the 3 dash hydroxyl ready to accept the GTP, sorry, ready to accept the GTP. The proper position of the correct DNTPs is also mediated by the lysine and arginine uh, on the finger domain, as the DNTP is also mediated and and DNTP is also mediated by this lysine and arginine amino acid on the finger domain, as they make weaker but important contacts with the beta and gamma phosphate of the DNTP. So now we are starting to see how these 3 residues are essential in this process. So uh, so that the stacking interaction is stabilized with the help of hydrogen bond between the 2 bases. Instead, if we had AT and G instead AT, or C instead G, the stacking would still work, but it 2 bases uh, it would fail to hydrogen bond with cytosine, and arginine and lysine will also not interact as efficiently, and these 3 DNTPs will keep getting rejected. So for a cytosine interact as efficiently, and these 3 DNTPs will keep getting rejected. So for a cytosine, only the DGTP works. All the stabilized interaction with the correct DNTPs is what stabilizes the close conformation of the DNA polymerase. Now let's see what is so spiral in this close conformation, especially in the close conformation. When this close conformation is triggered, the active site in the active site engage properly. This has to do with the divalent ion, And we also already have discussed about the divalent ion present. So now with the correct DNTPs at the palm region, one of the divalent mentioned that divalent ion makes contact with the phosphate of the DNTPs and other makes contact with the hydroxyl of the dotted strand to which the new DNTPs will be attached. All right, we have already discussed about the biochemistry of elongation. So simply moves into the palm. If the DNTP is correctly fit, the polymerase regarding the base pairing of the bases again is the finger domain. The finger domain which samples the DNTPs by stacking interaction and assess if the hydrogen bond is favored or not. The favorable bonding is then confirmed by proper contacts of the finger domain and the divalent ion. So the simple answer to this question is the finger domain dynamics. We already have discussed the biochemistry here. So in short, here you can see a close conformation. We are now, the next question is, how does DNA polymerase make phosphodiester bond? As we already know that to uh, the phosphodiester bond of DNTPs on the other side, the bond will be made with uh, the bond will be made with the growing daughter strand which has the free hydroxyl group. The reaction start with the divalent ion interacting with the DNTPs and the hydroxyl on the DNA strand. And for your information, these ions are not floating around in the active site. They are held together by a specific amino acid like aspartic acid residue in the palm region. So the mechanism is here. We can see the first metal ion. Suppose this is B. You can see it here. Interact with all this phosphate of the DNTPs, and this works to neutralize the negative charges of the phosphate, and it makes it inert. And these phosphates are usually named alpha charge, usually named alpha charge of the usually the alpha, beta, and gamma. And the other metal ion, let's call it as a ion, metal ion, that interact with a free 3' hydroxyl group and weaken the hydrogen bonding of the OH. Essentially, it works to take away the hydrogen. As a result, this oxygen here takes a radical form. So this oxygen is ready for the nucleophilic attack on the alpha phosphate of the DNTPs. When this occurs, the beta and gamma phosphate are released as a byproduct. And this bond that is formed between the alpha phosphate and the oxygen radicals is called the phosphodiester bond. So the process to make phosphodiester linkage involve a beautiful metal ion bond, metal ion catalysis reaction. So we can go back to the question discrimination against the DNTPs. If this DNTPs was instead of RNTPs, it would have it would have 2' hydroxyl on the sugars, and um, that makes the sugar a bit bigger, and the cavity of the active site where the sugar docks will not able to fit this RNTPs because of its bigger size, and this will further destabilize the staking reactions interaction if the polymerase try to work within it, and all these contacts that can leads to RNTP being destabilized and kick out of the reaction. So in this contrast, the DNTP discrimination by the finger domain and RNTPs are sampled by palm region. So now we have added the DNTP into the growing DNA strand. The D and DNA polymerase must move to one base to add next DNTP. So how does that happen? Let's just quickly complete. In the left hand side of the picture, the alpha phosphate and the oxygen together form the all important bond. After the bond is formed, the finger domain, the O helix or hinges back and takes away the phosphate. This pyrophosphate, this byproduct, is then um, released, and now the finger domain looks like it again looks like what it looks again looks before in the open configuration. Now, one hypothesis about the energy required to move the enzyme is possibly that the pyrophosphate release and the phosphate bond breaking provides the energy for the translocation of the energy enzyme. Turns out there is another prevailing hypothesis which involves the concept of linear diffusion, which state that because of this backward movement of the finger domain, the helix or the fingers act like a spring. So it short of a bigger picture, all that mean the finger act like a spring, and when the DNTP bind the finger domain closes, the configuration goes from open to close, which leads to the phosphodiester bond formation if the DNTP is correct. And after bond formation, finger domain retract back. So this movement or oscillation is similar to that of the spring, which forces the DNA polymerase to move with one basis copies at a time. Apart from palm and finger domain dynamics, each time the polymerase adds the DNTPs into the growing dotted strand, and the thumb domain responsible the template by some sort of rotational forces. The thumb domain detach for the short split movement and reattach in a new position. The exact mechanism underlying the movement are still being discovered, but researchers suggest that it is some combination of the spring-like oscillation and the pyrophosphate spring-like oscillation uh, due to the presence of this finger-like domain in DNA. So this is all about the elongation process, and we already well defined and well discussed the elongation process. So thank you so much for this, and we will in the next module we will discuss the termination process of DNA prokaryotic DNA polymerase. Thank you all.